today's topic of talk is telomere length, inflammatory marker, and oxidative stress in pre-eclamptic women. So, preeclampsia is a pregnancy-related syndrome, which is mainly by gestational hypertension with proteinuria. It affects 2 to 10% of pregnant women. In stage 1, there is a defective trophoblast invasion of maternal spiral arteries, followed by inadequate maternal blood supply, followed by placental ischemia and hypoxia. Further release of trophoblast microparticles into the maternal circulation. Then in stage 2, which is the systemic stage, the systemic endothelial dysfunctions happens in the form of vasoconstriction. That leads to hypertension. Then we have production of a lot of inflammatory cytokines, lead to X-aggregated inflammation. Then we have increased vascular permeability, leads to proteinuria and edema. Improper maternal spiral artery remodeling leads to decreased perfusion of the placenta. Hypoxic placental organ may release microvesicles, apoptotic cells, atheromatous plaques, and which promotes the X-aggregated inflammation, causing the increased amount of C-reactive protein, interleukin-6, TNF-alpha, oxidative stress marker, that is MDA, into the circulation. So what has been the gap analysis? And we are going to cover it. Like our study may provide insight into the pathophysiology of preeclampsia as well as its role of telomerase length, inflammation, and oxidative stress. We also found the disease severity can increase the BP and may also be considered for classifying preeclampsia. So with it, we have following aim. The aim is to find out the relationship between the telomere length, endothelin-1, markers of inflammation, and oxidative stress in preeclamptic women. These are the primary objective to estimate the telomere length in whole blood by using the PCR-based technique in both cases and control. Then quantify the gene expression of matrix metalloproteinase, MMP9, in whole blood by using the RT-PCR in both cases and control. Then quantify human endothelin-1 and inflammatory markers, interleukin-6 and TNF-alpha in serum samples by competitive ELISA. To find out, next is to find out the oxidative stress in the preeclamptic patients by estimation of MDA serum glutathione in both cases and control group. Our secondary objectives: to quantify the basic biochemical parameters like lipid profile, KFT, the liver function test, CRP, CBC by fully automated chemistry analyzer, and to compare and find the relationship between all the parameters. So these were our objectives. The material and method. So it's a case control study. The inclusion criteria for the cases: the woman more than 20 weeks of gestational age, new onset hypertension, which is more than 140/90 mm of HD, along with proteinuria. Then what is the exclusion? Like family history of preeclampsia, previous history of preeclampsia, tobacco consumption, alcohol consumption, irrespective of parity, and any other chronic diseases. Then we have divided the normal pregnant woman after 20 weeks of gestation. They were taken as control. So the first group was included the pregnant woman with the gestational age more than 20 weeks, preeclampsia with, of course, proteinuria and matched control. Further, this is the main group, and further this group has been subdivided into the following subgroups. The gestational age 20 to 34, that is early onset preeclampsia, and then more than 34 weeks, that is late onset preeclampsia, and accordingly the matched control. The third group subgroup is we had taken into account that we have divided on the basis of blood pressure changes. So control group included one having the blood pressure less than 139 by 89. Then all subjects were divided into three groups. A group were having the 140 by 92, 149 by 99 mmHg. B has 150 by 102, 159 by 109. C group has VP more than 160 to 110, means all 10 degree 10 mmHg difference they included. Then the whole blood sample was taken from all the subjects. Then their relative telomerase length, MMP9, interleukins, TNF, alpha, endothelin T1, MDA, GSTT, LFT, KFT, lipoproteins, and CBC. Everything was estimated from the blood sample. And this is the following statistical model we have taken into account by using the statical analysis. Then Shapiro-Wilk test, one-way ANOVA with Tukey Pastha, Crucial-Wallis H test, multinomial logistic regression analysis. This is how we have done the sample size. We have taken out the sample size as 100 subjects each group. It was calculated using the formula and Z power software for two independent variables. Methodology for relative telomerase length, we use the formula method given by Cawthon et al. That means whole blood was taken into EDTA. DNA was isolated, followed by nanodrop spectrometer. We did the quality and quantity analysis of the DNA. Then RT-PCR, telomere copy like T by S ratio we did. Then MMP9 gene expression, whole blood EDTA. In EDTA we have taken, and it was done by triazole method. Gene expression was done by RNA isolation, followed by quality and quantity measurement. Then river transcriptase used, we did cDNA synthesis, and followed by RT-PCR, we got the gene expression. Then MDA, GSH, endothelin 1, interleukin 6, TNF alpha was done by competitive ELISA kits, CRP, lipoprotein, LFT, KFT, CBC, all by fully automation method. So you can see the demographic data here of the first group, which included 100 in control and 100 pre-eclamptic subjects. So out of these parameters, the systolic and diastolic BP and proteinuria were significantly different, like raised in case of preeclampsia as compared to control. Rest of the parameters, no significant p-value were shown. Similarly, for RTL, MDA, excuse me. Simply for relative telomerase length, MDA level in controls and cases. So this is the graph, control versus cases. This is the graph for MMP9, ET1, interleukin 6, TNF alpha, C-reactive protein in control and cases. Light color is control versus cases. All values found significant. Then we did the correlation studies between the biochemical markers. So relative telomerase length showed a significant correlation with uric acid, C-reactive proteins, and then we have interleukin 6, then TNF alpha. Similarly, we found correlation with the positive correlation with MDA in control group. Then demographic data again of the second subgroup, in which we have taken the early onset preeclampsia and late onset preeclampsia. So 50-50 wise we had taken. So out of all these parameter, gestational age, there was a significant difference. Then the mean systolic BP and mean diastolic BP and proteinuria were significantly different among the control and the patients in both early onset and the late onset preeclampsia group. This is the relative telomerase length MDA level in early onset and late onset preeclampsia with control, comparison with control 1, control 2. Then GSH, endothelin 1, interleukin, TNF alpha, CRP level in both early and late onset with control. So we applied here Kushal-Wallis H test. GSH, this is the graph of endothelin, interleukin 6, then TNF alpha, C-reactive protein, etc. We found the significant difference, alterations as compared to control, as well as in both the groups, early as well as the late onset from preeclampsia. Again, this is the demographic data of the third subgroup in which we divided the subjects on the basis of VP changes. So A, B, C were the three categories. So this is the biochemical study. All parameters were evaluated for this VP group also. And this is the odd ratio calculation was being made. So let us move on to the discussion. The study of clinical parameters such as age, gestational age, and BMI are comparable in the group. Our study reports significantly reduced telomerase length in preeclamptic subjects as compared to control, as well as in early onset preeclampsia and late onset preeclampsia along with control. And the finding was similar to those of previous studies that was done by Byron Chantal T. et al. And Sukenik Halevi R. et al. Then subgroup based on gauge early onset preeclampsia and late onset preeclampsia along with match controls as well as VP subgroups showed reduced telomerase length in A and B group compared to the control. Odds ratio is 2 times lower than the control. So it is the risk factor for the preeclampsia. Then endothelin 1 level increased in preeclampsia compared to the control as well as in early and late onset preeclampsia along with the control group. So results of this study were supported by Tomimatsu T et al. And Singh et al. C-reactive protein level increased in preeclampsia compared to control as well as in both early and late onset groups along with the while comparing with the control, and the finding was similar to those of previous studies. Odds ratio here also is 2 times higher than the control. So MDA, interleukin 6, TNF alpha, HDL, uric acid levels increased in preeclampsia compared to the control group, both in early onset preeclampsia along with the control and as well as in late onset preeclampsia. Finding was similar to those of previous studies. GSH was reduced in preeclampsia compared to the control as well as in early and late onset subjects along with the control, and the finding was similar to those of previous studies done by M. Satish Babu et al. And Sardar Z et al. And this shows an association between the telomerase length, oxidative stress, and inflammation in preeclampsia as well as in subgroups. So preeclampsia is basically a state of oxidative stress because of hypoxic placental tissues, which may enhance the excessive production of oxidative markers and reduce antioxidant. Hypoxia is an inducer of stress and inflammation. Oxidative stress and inflammation will lead to endothelial dysfunction. Higher level of endothelin 1 may be attributed to alterations in production of anti-atherogenic proteins, angiogenic proteins, leading to endothelial dysfunction as a consequence of placental ischemia. So, endothelial 1 is activated by different mechanism through receptor inflammation and vasoconstriction. So we could not find any studies showing the association of this much large group of parameters, including the relative telomerase length, MDA, GSH, endothelin T1, interleukin 6, TNF alpha, and C-reactive protein, with an increase in blood pressure among the pre-eclamptic subjects. So our study found reduced telomerase length, reduced GSH, and an increase in MDA, interleukin 6, TNF alpha, endothelin C-reactive protein, uric acid, HDL level, with every 10 mm Hg rise in blood pressure in cases increased groups. So what is the conclusion? The current study concludes with an association of relative telomerase length, MDA, GSH, ET1, interleukin 6, TNF alpha, C-reactive proteins, and uric acid in pre-eclamptic patients. A positive association was seen for RTL, MDA, GSH, ET1, interleukin 6, TNF alpha, C-reactive protein, and uric acid level with increasing BP. Therefore, this study may provide a link between oxidative stress, inflammation, and with a reduction in telomerase length that leads to pathophysiology of preeclampsia. What are the future suggestions? We suggest, based on our findings of our study, like antioxidants and anti-inflammatory drugs may help in reducing the incidences of preeclampsia. Thank you so much.